We are almost near the end of the conference, and I don't know about you, but I am definitely feeling smarter. The only change I would have made is being the date that it is. I thought we should have had Star Wars as the walk-up music, but maybe we can plan that next time. Many of you in this room and watching have spent your entire adult lives peering into microscopes and other machines to see the tiniest inner workings in our bodies. And this is something you think about every single day. But consider this, the vast majority of people heard about RNA and messenger RNA for the very first time during the pandemic in the context of the novel COVID vaccines. As you know, RNA has been studied for half a century now with promise of broad applications for therapeutics. RNA can be the drug itself, or it can be the biological target within the body for a drug. So where are we heading now with RNA therapeutics? Today, we are speaking with 4 people whose life work involves different approaches to harnessing RNA to both treat and prevent disease. They have quite extensive CVs, as you know, with all the speakers, so I'm going to allow them to speak about their roles and their works in their answers. My first question is to Jeannie Lee. You have been researching RNA for decades in your lab at Harvard Medical School and at MGH. So you can start us off by talking about why RNA therapeutics hold such promise.
Absolutely. Thank you, Janet. So as Janet says, we've been studying non-coding RNAs for more than 20 years. And when we first got started in this space, RNA was viewed as kind of a boring thing. You know, it's a go-between between the DNA, which is our genetic blueprint, and the cellular workhorse, which is considered at the time to be the protein. But all of that has changed in just 20 years. So we now know that RNAs are a thing unto themselves. So for instance, the Genome Project has shown us that 98% of our DNA doesn't actually encode for a protein, but instead makes non-coding RNA. And it's estimated that there could be probably upwards of 10 times as many distinct forms of non-coding RNAs than protein-coding RNAs. And the thing is that the pharmaceutical industry has been kind of slow in catching up to this idea. So my understanding is that commercially available drugs and a lot of the pharmaceutical effort right now is being directed at just 700 proteins or genes that make proteins. And that's a really small sliver of our total genetic space, way less than 1% of what's encoded in our DNA. So the idea is that if we could actually match up these non-coding RNAs that are being expressed in huge numbers across their genome, and we could target those non-coding RNAs, then we would vastly expand the chemical space in which we can design drugs. And so that's what many of us here are trying to do. And as Janet says, there are 2 different approaches to thinking about RNA therapeutics. One is RNA as a drug itself, and the other is RNA as a target for some other kinds of drugs to hit. And so we've been doing both, a little bit of each. And so Translate Bio, which we started with MGBV many years ago, has been acquired by Sanofi, use messenger RNAs as vaccines and also as drugs to treat metabolic disorders such as ornithine transcarbamylase deficiency. And in our own lab, we're now using non-coding RNA as a target. We're designing some other kind of drug to hit non-coding RNAs to treat neurodevelopmental disorders.
Let's go from your lab to another company. Sepp-Lorenzino, your company Intellia, is the first to have a clinical in vivo proof of concept for CRISPR gene editing. Now, what can you share of Intellia's latest developments? You are using an LNP with RNA cargo for in vivo gene editing.
Thank you, and thank you for the opportunity to participate here. So yes, at Intellia, we have demonstrated very clear clinical proof of concept of CRISPR as a new class of medicines. For that, we have used a lipid nanoparticle similar in design, of course, to what you have received as a jab for COVID vaccine. Of course, there are differences in composition, formulation, and cargo, but rather than having an mRNA as a vaccine, what we put inside that LNP is a messenger RNA that codes for the CRISPR, the gene editing machinery, alongside with a guide RNA that's a synthetically made single-stranded RNA molecule that's going to tell that Cas9 protein, that gene editing protein, where to go in the genome to introduce the edit. So we have used that technology now in over 15 patients. Data has been disclosed for our lead program, NTLA-2001, for transthyretin amyloidosis. And what we have shown is that you could see a very well-behaved, dose-dependent pharmacokinetics, pharmacodynamics that leads to the intended therapeutic effect, which is to knock out a gene that codes for a toxic disease-causing protein called transthyretin in a disease called transthyretin-mediated amyloidosis. With a gene editing event where we're turning a gene off, once that happens, there is no normal mechanism for the cell to undo that knockout event. So once that the gene is off, it's off for the life of that cell. What we have shown in our preclinical data, and now we are gathering human data, is on the duration of the effect. We are expecting that once that these edits are introduced, the persistence will be long-lived, potentially lifelong. And this is where we begin to think about the opportunity to intervene early in disease with very long-lasting, again, potentially once-in-a-lifetime type approaches. As we are applying a platform approach, this strong clinical proof of concept de-risks what we're doing in other pipeline programs in vivo, whether it's to knock out, again, disease-causing genes, or to make changes to insert genes for loss of function diseases. In addition to our in vivo platform, we're also using lipid nanoparticles as a tool that allows us to work with cell therapies, to be able to introduce as many gene editing events, changes as are required for making those cell therapies have all the properties that we expect for them to be safe and lead to efficacious and durable responses.
All right, thank you. We have the CEOs of two companies to my immediate left who are taking different approaches to RNA therapeutics. Sarah Boyce, you are the CEO of Avidity Biosciences, which is working on ways to deliver RNA to specific tissues in the body outside the liver. How does this delivery system work?
Yeah, so one of the things in the RNA field today, there are several RNA therapeutics approved, and they've made really transformative differences in people's lives, but they all target the liver. So when Avidity was founded, our goal was, how can we tackle one of the fundamental challenges in the RNA therapeutic space, which is delivery? And we do that through a really simple way. In using what we all know well, which is monoclonal antibodies, which are very specific. So we actually conjugate our RNA, either an siRNA, or we're also using a PMO for Duchenne's muscular dystrophy, but primarily siRNAs. So we conjugate our RNA to an antibody, and the antibody is effectively the delivery vehicle that gets our therapeutic to our target tissue and cell type. Where we started is using an antibody for the transferrin 1 receptor, and that allowed us to be able to deliver to muscle cells. So today we have our first program in the clinic for myotonic dystrophy, and the goal is by the end of the year to actually have 3 programs in the clinic for 3 different rare diseases, all of which where we're targeting muscle cells. But we're able to do much more than that, because if you think, well, that's with one antibody. We also have identified other sets of antibody receptor pairs that allow us to deliver an RNA therapeutic to immune cells. So, for example, to B cells, to macrophages, We're able to target the heart. So we can really look at building on the incredible work that's been done in taking RNA therapeutics to liver. And what we can do now, or our goal, is to be able to really change all of that by delivering RNA therapeutics to other tissues and cell types. It's a really simple concept, using an antibody. When I first spoke to the company, I was like, why has no one thought of that before? It's actually pretty difficult to do and took a lot of engineering to get us to where we are today. But it's really exciting to be able to, in rare muscle diseases, there are people who have been living with these diseases, ideal for an RNA therapeutic approach, and no one's been able to deliver there. So it's a hugely exciting time for us to expand on the work that others have also done.
And remind us, your company recently went public during the pandemic, correct?
We did. So we went public in June of 2020. So we were one of the first companies to go public in the midst of the pandemic. Really interesting experience. And I remember when you're taking a company public, you do drafting when you're drafting the S1. So I was talking about it last night, actually, with some colleagues. And we all got on this drafting call. And you have an army of people when you IPO a company. So there must have been like 60 people on this drafting call, all of which none of us had ever drafted an S1 over Zoom. So our first call was an unmitigated disaster. It really was. It was just awful. People couldn't use the Zoom properly. And then we actually found our groove and found it to actually be incredibly efficient. And I think, in speaking with the investor community, that was also the same. And actually led ultimately to a really big influx of capital into the biotech industry. But it was certainly a unique experience.
Yeah, and definitely those investors are learning a lot about RNA right now.
Yes, they are.
Jim Burns, at Locanabio, you are doing something very different. You are looking to target RNA. And how are you doing that and why? You are, of course, an RNA targeting gene therapy company.
So thank you, Janet, and hello, everyone. And actually, I want to start off by saying I'm the only private company up here, and I'm glad that I'm private in this current environment. But what we're doing, which is very different in terms of how we're attacking disease-causing RNA, sorry for my voice, is that we are using payloads that specifically and naturally actually target RNA. And we can engineer those payloads and deliver them via gene therapy, so we can get a durable response with a single administration. And we can engineer these payloads to manipulate disease-causing RNA by different ways. So they can destroy the RNA, if that's what we want to do. They can just block the RNA. They can affect exon splicing. And we can also, because of their size, which are quite small, relatively small, they fit into a single AAV vector, include additional guides or means by which they can do what's called multi-targeting. So if you have multiple transcripts that you would like to target, we have the ability to do that. And, or within a given exon, if you wanted to target multiple exon splicing enhancer sites, you can do that as well. So it gives a lot of flexibility in terms of the types of disease-causing RNA we can address. The other thing that's very interesting about our platform is that for certain diseases, it makes sense if you can target the mutant allele, disease-causing RNA, and not the wild type. And our approach has the ability to do that, and that's particularly important for triplet or repeat expansion diseases, and there's many of those, at least 50 of those types of diseases. So there's lots of different ways of trying to target RNA, and I just want to also say is they all have a role, and what we're all trying to do is ultimately try to provide a toolbox for the future where you can look at what's the problem that you're trying to solve and what is the right tool to bring to bear. And sometimes that's going to be a gene therapy, sometimes it's going to be something for targeting the gene itself and affecting DNA modification, and other times it's going to be targeting the RNA. So we think our approach is going to have a lot of adaptability and flexibility for adding additional tools to the toolbox for being able to do that.
tools to both use RNA and in some cases you said destroy RNA. So back to Jeannie, as Jim said, he is targeting toxic RNAs among the many things his company is doing. You are looking at ways to target a wide range of RNAs. Tell us about the research happening in the lab right now.
Right. So, sometimes we want to target these toxic RNAs that are causing neurological diseases, but sometimes we simply want to target RNAs that play a normal, healthy role inside cells. And so that's one of the things that we're doing now, and I'll just give you one example of it. For instance, we're targeting an important long non-coding RNA called EXIST, which is responsible for silencing one X chromosome in girls or in females. And what's interesting about this inactive X chromosome is that it's basically like a big bag of spare parts, if you will, a lot of spare genes that we carry around for the rest of our lives to no purpose at all, or at least not to any normal purpose. So what we're trying to do, and the reasoning behind this, is that since there are 1,000 good genes on that inactive X chromosome that never get utilized, why not try to reawaken a select number of those X-linked genes to treat disorders that are caused by a mutant allele on the X chromosome, which is active? And so that's the X reactivation platform. And we're thinking about this for various neurodevelopmental disorders, including Rett syndrome, Fragile X, CDKL5, many of them on the that's located on the X chromosome. And the nice thing about the neurodevelopmental sphere is that the approach that we're taking, which is using an antisense oligonucleotide directed against these non-coding transcripts, is that the brain happens to be a privileged compartment where it is not necessary to use a delivery vehicle to get antisense oligonucleotides into brain cells. And so that's where we're starting, and hopefully we'll succeed in the near future.
I want to ask Laura Sepp-Lorenzino a quick question. You talked about the neurodevelopmental diseases. For the therapy that Intellia is doing, can you remind us what diseases you are targeting now?
Yes, so in vivo we are targeting, because we're using an LNP, a lipid nanoparticle, as the mean of delivery, we are targeting diseases that originate for genes that are in the liver. So our first clinical candidate is targeting transthyretin, which is the causative agent for transthyretin amyloidosis. That is closely followed by another clinical program that's targeting a key pathway element in a disease called hereditary angioedema. The goal there is to restore control in a pathway and prevent the disease, inflammatory events, edema events that happen in hereditary angioedema. In addition to that, as what's disclosed of the pipeline is using CRISPR to insert genes. So we have the name programs. We have two with our partner Regeneron, hemophilia A and hemophilia B. And then we have a wholly owned program for alpha-1 antitrypsin deficiency, where we're both inserting a healthy copy of the genes in patients who have a defective mutation, as well as to using a knockout approach to take down the expression of the harmful mutant allele. And then on the other side of the pipeline, which is our cell therapies, there we have a hematopoietic stem cell program with Novartis, where Novartis is the lead, and wholly owned by Intellia is our program NTLA-5001, which is a TCR, T-cell receptor, T-cell mediated therapy for acute myelogenous leukemia, and that is a clinical program. There was a presentation earlier this week at Keystone where we unveil more details about our next ex vivo program, NTLA-6001, where we're using our allogeneic platform for CAR Ts, starting with a CD30 targeted CAR T. And in addition to this, we continue to build our capabilities. When we talk about Intellia, we consider ourselves a full spectrum company, not just because we're doing in vivo and ex vivo, because we want to be unlimited by our ability to deploy the right gene editing modality alongside the right delivery modality so we can tackle any diseases. And for that, we continue to build on our gene editing platform, having two different Cas enzymes, one that works beautifully on an AAV, the other one that we're using for LNP in the clinic. We have a base editor, we have a DNA writing technology, and we continue to push on both editing and delivery.
I'm going to ask Sarah and Jim also what diseases your companies are targeting in a moment. But first, we have some good questions coming in. I think this one is for Jeannie, but everyone welcome to weigh in after she answers. How can we build a better understanding of what role non-coding RNA plays in specific diseases?
That's a great question. So I alluded before to the fact that 98% of our genome is non-coding, and it turns out more than 90% of that space is transcribed. So there's a big debate in the field right now about how many of those 200,000, maybe up to 1 million transcripts are truly going to be functional, are going to play a role in disease. So one thing that I will say is that these GWAS variants that have been identified, the vast majority of them are actually out in this non-coding space that are producing RNA, or non-coding RNA. They're not in the protein coding genes. So I think the knowledge gap is going to be how many and which of these non-coding RNAs are disease-related, and then secondly, how can we target those non-coding RNAs? So there are delivery issues that have been mentioned, but also what modalities are we going to use? Are we going to use siRNAs, aptamers, ASOs, or can we also design small molecules to target these long non-coding RNAs? And I think, being bullish on this, I believe that the answer is going to be yes, and we recently had some proof of concept that exist RNA, for instance, can be targeted by a small molecule with phenotypic impact on X and activation.
Sarah, I'm wondering if you can answer this question before we talk about the specific diseases you target outside the liver. This question coming in, what strategies are currently being explored by academia or other early stage researchers to expand the tropism of LNP delivery beyond the liver, excluding direct administration?
Yeah, so I'm not sure I'm the best person to answer that, in that we're actually not using LNPs, so that is not in our technology. We're using an antibody conjugated to an siRNA or a PMO, and we can also do ASOs as well. There are lots of other people also working on delivery, but I wonder if maybe one of my other colleagues, actually you're probably best to...
Or I think also Laura and Intellia, because you're using the LNP for delivery.
Exactly.
Yeah, so LNPs work beautifully for liver, right? And actually, they have a number of advantages, for example, over viral delivery systems, being non-immunogenic, being able to precisely dose to target, redose as needed, and then you're unlimited by the cargo capacity, right? So you could put, large enzymes or combination of nucleic acids inside the LNP. It works well for tissues that are well irrigated, that there is a good blood supply, but also that there is the ability for particles to leave the blood supply and enter into the tissue, with liver, being the prime example. You could also look at other fenestrated tissues, looking at spleen, bone marrow, as well as you could administer by local administration. So an example for Intellia, again, as we're pushing on delivery, we're very keen on identifying and developing lipid nanoparticles that can in vivo deliver to hematopoietic stem cells. Right now, as you know, there is beautiful data on taking hematopoietic stem cells out of the body, modifying them for sickle cell disease, and then infusing them back into the patients. The limitations with that is, it's a complex manufacturing, it's a bone marrow transplant, so, for the patients, there is, morbidity associated with that, cost, complexity. Our vision is to be able to do that by a single, outpatient infusion, where now the lipid nanoparticles will go to the hematopoietic stem cells in the bone marrow, introduce the edit, whether it's for sickle cell disease or for other inherited blood disorders. So we're, actively working on making that a reality.
Think about that, a single outpatient infusion. Okay, Sarah Boyce, back to your work at Avidity Biosciences. What diseases are you looking to target first?
Yeah, so initially we're looking to target a range of previously untreatable rare muscle diseases. One of the elements when you have a technology like ours, there's a lot of places you can go. And we were initially directed with muscle because of finding how well the transferrin antibody worked with regards to delivering RNA therapeutics to muscle cells. So then from there, so you've got muscle. Where do you then go? So in the RNA space, obviously you're looking at genetic diseases. And one of our other sort of goals as a company is to be able to make a transformative difference in people's lives. And part of that aspect in doing that is looking at diseases that were previously untreatable and where there is a very high unmet medical need. So our first program in the clinic is for a disease called myotonic dystrophy. It's a rare genetic disease. Estimated in the US, there's about 40,000 people living with myotonic dystrophy, and they have nothing. It's caused by a triplet repeat that ultimately means that the RNA kind of gets stuck, and that impacts a whole range of downstream splicing that's needed for effective muscle function. Our next two programs, one is in DMD, and in DMD there are approved treatments in DMD, but they're very specific to certain exons. We're targeting exon 44. We went there because no one was looking at exon 44. It's one of the rarer mutations for these boys. And then our other program, that kind of hand in hand, is for a disease called FSHD, which is facial scapular humeral dystrophy. The name is from where there's loss of muscle function over time. It's actually caused by an inappropriate and an aberrant expression of an embryonic gene called DUX4, and when that's expressed, the muscle very rapidly starts to lose function. And there we're looking at a straight knockdown approach of knocking down the expression of DUX4. We have additional programs in the muscle space. We also have earlier programs in immunology, and also early programs looking at being able to target the heart and being able to target a set of rare genetic diseases in the heart. So there's a lot of applicability as to where we can go. And I think for us, the guiding point as to where we go with regards to diseases, we look at two things. One, is this applicable to our technology? Do we think we can make a profound difference? And then secondly, how high is the unmet medical need? And I think with those two guiding points, from a strategic perspective, that will always guide us to being where we can make the most impact for the patient communities that we're trying to serve.
A broad range for a young company. Jim Burns, the same question for you at Locanabio. Which diseases are you looking to target first?
So we're focused in the CNS, neurodegenerative type diseases, as well as neuromuscular, just very, very similar to what Sarah is doing at Avidity. The repeat expansion diseases are replete in some of those areas. I mentioned, I think there's probably at least 50 repeat expansion diseases, probably more than that. And what's interesting about the repeat expansion diseases is that the RNA forms a structure with these large triplet repeats that ultimately causes the toxicity of the disease. And our approach can target those structures once they get beyond a certain size, and allows us to target those structures in a allele selective way, which I mentioned before. And one of the diseases that this is really important for is for Huntington's disease. And there's no current therapy for Huntington's disease. It's a horrible disease. And what we're doing is developing an approach that targets these repeat expansions and will knock down the disease-causing RNA and spare the wild type. And what we've learned recently, unfortunately for patients in clinical trials, is that it seems like it's a good idea to spare the wild type to make sure that you preserve it. And so that's an example of the approach that we're taking. And another very interesting neurodegenerative disease is smaller. Huntington's disease is similar in size as DM1 that Sarah was talking about 40,000 patients in the US. Another interesting disease is C9orf72 ALS. It's the most common form of familial ALS. And it's also a repeat expansion disease, but it's a hexanucleotide repeat. And what we're doing there, again, is targeting the repeat. But what's really interesting about this disease is that both the sense as well as the antisense transcript are toxic. So to be able to treat this disease effectively, you have to have a construct, in our case, an AAV construct, with a single product that can target both of those. And so our approach is doing that. We can do it with a single product. We don't need 2 ASOs or 2 siRNAs. And we can also do that in an allele selective way. So those, by way of example, Janet, are two of the things that we're really excited about. And they're both repeat expansion diseases. But our technology can also be used for other types of diseases, such as splicing modification. And one of our favorites there is for Duchenne's muscular dystrophy. And so more to come on that. And I do want to put a plug in that we have an oral presentation at ASGCT next week on our C9 work, and we're really excited about that.
Duchenne is, I call it the disease that has an elegant name for the most horrible, horrible condition. So hoping that we find a cure for that. This next question, I love it so much. I actually invite the person who submitted it to raise their hand if they so choose. I think I'm going to have Jeannie answer this first, but all of you can weigh in with a yes or no. Do we now need an RNA human genome project analogous to the DNA genome project?
Yes. Where do I start? Yes. So many people are talking about this right now. In fact, the NIH is convening an RNA-ome workshop in a couple of weeks. I'll be participating in that to discuss where do we go next with the RNA research. Now that we understand that RNA plays so many different diverse roles, not just as messengers, these go-betweens, but as entities in themselves, actually being the workhorses of the human cell. And so there are a number of different places that we should be thinking about. We should think about alternative splicing. We should think about ways in which RNAs can be modified chemically, naturally, by human cells. So RNA methylation is just one such modification. And then what do those modifications do to the function of the RNA or to the stability of the RNA? So there are many, many different, to answer your question, many different ways in which one could proceed in studying RNA.
I assume that you all agree that such a project would be worthwhile. Maybe it will be launched here from this meeting if people are not already trying to do this. So we all look for bright spots from the pandemic, and as I mentioned at the very top, it was the first time that you had average non-scientists talking about RNA and trying to figure out what it was and trying to understand this in context of the vaccine. So I want to ask you if the pandemic and the attention on RNA and messenger RNA has accelerated the progress in this field and in this science. Laura, let's start with you. I know Intellia has been at this for many, many years before the pandemic, but what sort of acceleration have you seen?
Yeah, no, I think that there is, first it's become a word that, all of our families talk about, right? So that comfort that this is a technology that works. Second, I think it's the demonstration of the versatility of the modality, and then helping with manufacturing. I think that in the beginning there were maybe some limited options for large scale-up of mRNA, which now it's commoditized. So, that and some of the analytical development that needs to go along with that has been, accelerated by the mRNA vaccines.
Jim, I'm asking you that question.
Yeah, it's really interesting because, I started at Locanabio in December of 2019, and we were planning about, when are we going to do our next raise? And you would think that it would just drive you nuts, the fact that we then just 3 months later get hit with COVID, which it did, but it also afforded an opportunity for us, oddly enough, because all of a sudden the activation barrier for people to be excited about mRNA was much lower. And so we were able to close a Series B in December of 2020, 100 million Series B, in part because people knew RNA was important. We had great science behind what we were doing as well, but I just think that it made it easier to have the conversation about why RNA is important.
So Jim, you raised that massive 100 million, and Sarah, of course, your company, as we mentioned earlier, went public during the pandemic. So now as a newly public company, talk to us about the acceleration that you're seeing in your field.
Yeah, and I think that, to the points my colleagues made, the excitement and understanding around the RNA therapeutic space, in turn, like when we IPO'd, we were the largest ever preclinical IPO. I think we're still like the second largest ever preclinical IPO. What that influx of capital enabled us to do was to be able to actually accelerate our programs faster than we otherwise would have been able to. So actually we were looking at our S1 a couple of weeks ago, and if you look at the commitments that we made in the S1, we either hit them or have exceeded and gone faster because of that influx of capital. And then we did a follow-on raise last year that also went extremely well and puts us in a really good position now of having the cash that we need to be able to get our drugs into the clinic, expand our pipeline, and hopefully be able to deliver on the promises we made, which was to get RNA therapeutics outside of the liver and make transformative differences in people's lives. But I also remember at the time, just when we'd started our drafting, and it was in the midst, we all remember the early days of the pandemic, and people were scared, and we didn't know what was going to happen. It was like, should we be doing this? And it was like, no, let's just keep on, and we'll see where we are when we get to the point where we're ready. Thank God we did that, because at that point then, one of the great things about our industry and our community is we work stuff out. And we find a way to innovate and find a way to actually make things happen.
And as was mentioned even yesterday in many of the panels, even during the market turmoil and the economic stress, it's always a time for biotech to accelerate.
Absolutely.
We are down to our last 2 minutes, so I want to do a very quick, get a quick answer from each of you, but I'm going to end with Jeannie because she does the most lab work right now. And that is, with this acceleration, where do you see us, Sarah, 25 years from now?
Wow. I think the opportunity for people with genetic diseases to actually have treatments, be it through the range of different approaches that we've seen,
Jim?
that builds a better world.
Yeah.
Yeah, no, I think it goes back to what I was saying before. We're going to have an awesome toolbox for genetic diseases. And a big part of it is going to be delivery, what Laura was talking about. We have to have a better delivery of our payloads, whether it's LNP or better AAVs. But that's what I see in 20, 25 years.
Yeah, and for me, it's precision medicine, early intervention, right, before the disease sets in, ability to change people's lives.
Yeah, and for me, I think we're finally going to get beyond the 700 proteins that everybody targets and expand out to tens of thousands, if not more, RNAs that are associated with disease.
And so you have today, during these 40 minutes, been able to peer into the future with all of us to see the promise of RNA therapeutics, hopefully to treat the diseases that currently have no treatments or cures. I really want to thank our panel for this fascinating conversation. It zipped by so fast. I know 12 more brilliant people are waiting to come on the stage. So thank you very much, and thanks
Thank you.
for your great questions too.